So session 5 is links between reductions in deposition and climate resilience. And we are going to start with a talk by Todd McDonnell.
Hey folks, thanks very much NADP for having me, and thanks for you all for being here. I'll be giving an overview of 3 different projects that we've been working on. So there's quite a few co-authors here. I'll be talking about the effects of climate change and air pollution on soil and vegetation across the United States. So we haven't really quite gotten to the linkages yet, but so this is really quite similar to an extension of the critical load session yesterday. So each of these projects are at different stages. The first project I'll talk about relates to soil acidification critical loads, where we compared steady state and dynamic model results. The manuscript is under review, so we pretty much wrapped that one up. The understory work is in progress, but we've come a long way on that. And the tree response modeling work is in its early stages. So here's some just background on the 2 main types of soil acidification models that we use for generating critical loads. We have the steady state and dynamic models. So the steady state models are relatively simple. There's a low set of input data requirements. They can be used to characterize broad extents, and we generate critical loads from those models. And there's really no time frame for recovery specified. It's just it's under a steady state condition. So you can see the map there. That's a map of critical loads using the steady state model across the United States, forested areas only. For dynamic models, those are relatively complex. They require field data to be collected, in particular soil and stream chemistry measurements. So we typically use those to characterize sites at which we have those data available. So we call those, when we generate critical loads, we call them target loads, but we consider it a type of critical load because the target load is to attain the specified endpoint by a specific point in time. So the dynamic models can simulate change through time. That's why we call them dynamic. So the objective of our study was to compare critical loads derived from steady state models with target loads derived from dynamic models. And so we have one study where we have steady state critical load results across the whole US. There's that on that previous slide. And those are shown by those gray dots. It actually looks like a gray mass, but those actually are individual points at every forest pixel across the United States. So we paired those steady state model results with the nearest neighbor critical dynamic target load model result. And then we used those paired values for comparison. And we did that across, so we have multiple studies with dynamic critical loads. Since we have studies in the Adirondacks, studies along the Appalachian Trail, one study along the Appalachian Trail, and then the light blue dots, those are derived from a steady state model, but it was informed by a dynamic model. So we included that as critical loads informed by a dynamic model. So here's an example comparison between target loads and critical loads for 97 Adirondack Lake watersheds. So we see much lower target loads from the dynamic models relative to that narrow range of critical load from the steady state models, up around 10 kilogram of sulfur per hectare per year. And I should mention that these are critical loads of sulfur only for acidification. And these differences are really mainly due to the specified soil criteria for recovery and the time frame to recovery. So for those dynamic model results, those are generated based to attain a pre-industrial base saturation condition by the year 2150. And so not quite pre-industrial, we were within 3%, we were trying to go within 3% of historical base saturation. So with those dynamic models, we can get that estimate of the pre-industrial year 1850 soil base saturation, and then we can set our target to try to attain near to that condition by a specific year. In this case, we use 2150, so about 130 years from now. Whereas with the steady state model, that was done to attain a soil solution base cation to aluminum ratio of 10 under a long-term steady state condition with no real specified point in time for recovery. So really it's the requirements of having to recover to a pre-industrial condition by a specific year is what largely drives that lower target load relative to the dynamic critical load. And you'll notice the box plot for the dynamic target loads extends all the way down to 0. Those are lake watershed soils that we call them can't get there from here. There really is no level of deposition that we can reduce to in order to attain the specified chemical criterion. So this, it's important for understanding the trajectory of these watershed soils when you're looking at aspects of vegetation growth and health. So this is the spatial extent of the critical load exceedance when you use the steady state model results on the left or using the results that were either derived or informed by a dynamic model on the right. So we look at the spatial picture of steady state critical load exceedance. There's very few sites that are in exceedance, but when we are swapping in the dynamic model results, there's quite a bit more exceedance due to those lower critical loads. So fair amount of exceedance remaining in the Adirondacks, throughout the southern Appalachians, and along the Appalachian Trail. So our recommendations on this, from this, was to use the dynamic model-derived target loads where they're available. Feel they're more robust, but it's not to say that the steady state results aren't useful. So results from both types of models are still useful for resource management and evaluating emissions policy. Even if we don't have dynamic target loads in an area, the steady state model can still give you an indication of whether or not you are in an acid-sensitive area or not. So moving on to the understory response modeling work, where we developed critical loads of end deposition, and we also evaluated climate effects. So just a quick slide on why we're looking at the understory. These species, they play a significant number of ecological functions and also provide a variety of ecosystem services. So just kind of want to show this, it's kind of, some people, often, we can overlook these smaller organisms in the forest when we're out there. But they provide a number of functions and services that we enjoy. So we use the PROPS model for the analysis, which is a logistic regression model based on 9 predictor variables. So the variables we use in the model are aspects of climate, deposition, soil characteristics, and light availability. And so it's a 9-dimensional model, and we can use that. The model predicts the chance of occurrence, the probability of occurrence. And so we fit the data to presence-absence, observed presence-absence of the species across a large number of plots. And so our results are somewhere between 0 and 1. What's that probability that we expect the plant to be able to occur in a specific location or a specific point in time? And so our first regional case study was for Great Smoky Mountains National Park. And we identified characteristic species for each of these mapped vegetation classes that occur throughout the park. And we estimated changes in the occurrence probability according to scenarios of nitrogen deposition and climate change. We focused on air temperature increases. So we calculated critical loads of end deposition to protect the present day occurrence probability, and that's an important point there. The critical load is relative to a state, and it's relative to protecting the chance of occurrence that we expect the plants to have today. So here are maps of just an example of critical loads to protect 90% of the characteristic species within each of the vegetation classes, and also the exceedances. So so each vegetation class has somewhere maybe 10 to 30 different species that we consider to be characteristic of that area, and so these critical loads protect 90% of those species. So you see, sorry for the small font there, but those red areas are critical loads less than 2, and then the orange area in the northwestern portion of the park are critical loads of between 2 and 4. So really generally find exceedance where we have the low critical loads, which makes sense. That's just an example. We can set critical loads based on different points. We can go to the most sensitive species. We can look at the median, the critical load based on the median, the 50% of the species. So we can look at different points in the distribution. But just for an example, just chose the 90%. And so we're currently working on extending these props applications across 8 focal park units, and that's being written up as a report right now. And then we're also developing a database that will include results for 192 additional parks. So we'll have critical loads in that database. We'll also have these changes in the occurrence probability. Well, I'll show those on the next slide. But the plot here shows distributions of critical loads across the vegetation classes within each national park. And this is based off the median critical load. Like I showed the 90th percentile on the last slide. This is the median. So this is protecting at least 50% of the species, or 50% of the species. So those distributions, those lower distributions that are below that 5 kilogram N per hectare per year, those are occurring in Glacier, Grand Teton, Rocky, and Yosemite National Park. So these higher elevation parks tend to have lower critical loads. This is a plot, this is the change in occurrence probability. So the zero value, zero change is right down the center of the plot. And so decreases in occurrence probability extend to the left, and increases in occurrence probability extend to the right. And those are the 8 national parks on the left side of the plot. And so the scenarios here are increases in air temperature, 0.5 degree, 1 degree, and 3 degrees. And so he sees, this is based on the most sensitive species. So these are kind of the extreme case per vegetation class within each park. And so we see some significant declines in occurrence probability with increases in air temperature, some of which go to the point of 100% decrease, so expected to be near complete extirpation of the species in those locations, and a lot of responses in that 50% reduction range. And there's a few species where there's a few map classes where the most sensitive species actually still increases, so there really are no negative responders in that map class. So moving on here to the tree response modeling, where we're working on developing revised models based on some of the work that you saw yesterday from Linda and Chris, and evaluating air pollution effects, climate change, and also agents of disturbance. This is just going to be a quick kind of overview because we're still in the early phases of this project. So this is kind of just a preliminary look. But the responses that we're concerned with here are growth rate and survival, as you saw yesterday. But we're also considering aspects of recruitment as well. We'd like to. It's not currently scheduled, but that's something that we'd like to do. And we're using the FIA database for this. We have more than 20 years of data now. We're revisiting, repulling data from that database relative to what Horn used. And we're also expanding the set of candidate predictor variables. We have all these aspects of air pollution. Climate is an important factor. We want to look at those interactions between deposition and climate. But also a really important factor here that hasn't been considered yet are these agents of disturbance, more acute agents of disturbance like insect infestations, disease, and fire. We used the previous set of models in the Intermountain West to evaluate species response and wrote that up in a report. And a lot of the feedback we got was, these are Western parks where a lot of these disturbances are quite prevalent. And so a lot of the feedback we got was, where's the disturbance? Where are these acute disturbances? And we had to say, well, they're not in there. And so this is a really important part of this modeling to be able to include these aspects of disturbance, to be able to have a fuller picture of how these trees are responding to different drivers. Light availability, also very important for tree growth. Some of that has been brought into play using these competition metrics, but we're going to specify that directly in the model. And also various aspects of soil characteristics that are available now nationally with these Surgo and StatsCo databases. So in terms of the modeling framework, we're considering machine learning, and also there's interest in exploring Bayesian methods as well. And we have, right now we're looking at 10 focal species, but once we get through the whole database, there should be enough data for at least 100 or so tree species. So in terms of the research questions here, what are the most important drivers of tree growth and survival? Just starting off broadly, let's just take a look at all this. And for some species, maybe some of these drivers are more associated with some of these acute disturbances that I mentioned on the previous slide. And which trees show increasing, decreasing flat responses to air pollution and climate factors? So this is extension of the previous work. And that folds right into critical loads. So what are those thresholds of air pollution that relate to specified levels of effects on tree growth and survival? And also the same kind of thing for climate change as well. Is there a threshold of a change in climate that is particularly, is it associated with a particular level of effect? And really, to try to dig into these data a little bit more, I mean, how are these drivers interacting with each other? How do they interact to affect the growth rate and survival? I think it's an important question, and that kind of leads into, how does air pollution affect the likelihood or severity of one of those acute disturbances? I mean, do we see greater extent of insect infestation or mortality associated with an insect outbreak in areas that have high nitrogen deposition? I mean, some of those, there's studies out there, gypsy moth, that show that, but it hasn't been done at a broad scale across 100 tree species. So, yeah, this is sort of just the kinds of things we're interested in, not all the questions, but just some of the main questions. So with that, I'll take other questions. If anyone's...
Thanks for a great talk. Going back to the soil acidification critical loads, my question is, do those models take into account base cation deposition? And if not, kind of how could that be incorporated and how might it change the results?
Yeah, they definitely take in base cation deposition. Really, that simple mass balance model is a base cation balance. So it's the base cation deposition and the weathering, and then you subtract off, an uptake and removal term, and then also an allowance for some cations remaining in the soil to protect forest health. Yeah.
I have one online question.
Sure.
Sure. All right. This question from Jim Renfro. Is it possible to add 5 to 10% increase in precip with the increase in temperature with your nitrogen and climate scenarios?
Yes, we definitely could do that. Yeah, when we looked at scenarios in the project, we focused on the air temperature changes, mostly because when we look at some of the climate forecasts in the eastern U.S., is that what's going to happen with precipitation is very uncertain. Sometimes, some cases it's shown to increase or decrease, and so we just left that alone and just focused on the air temperature. But we could definitely, we have a precipitation term in there, and we could definitely make changes to that and look and see how that plays out.
Okay, so we are going to move on now to Sean Wetmow from the Ontario Ministry of the Environment. Did I butcher your name?
No, you butchered where I come from. That's fine.
Okay.
That's fine.
Right and left arrow.
Okay. Apparently, I've just changed positions. My co-author is the Ontario Ministry of Environment. I'm Trent University. So I'm here to introduce Coffee Break, which is in 20 minutes from now, so hopefully that. And I have been down this part of the woods about 20 years ago with 5 Shropshire sheep and a 6'4" American chewing tobacco. So I've got a long story over beer. So hopefully I'll get through this in 15 minutes or so, and the take-home message is right there. So if you do have a bit of a nap and wake up, you've probably not missed too much of the talk. As we've heard, both yesterday and actually just now, the nitrogen cycling environment is very complex, and we know it affects ecosystems in a variety of ways, and we need to manage nitrogen deposition to protect the environment. And we've heard a lot about the critical loads and the way they are generated and used. And there's kind of 2 ways you can do it. One is from empirical observations, where you have data and you predict either growth or occurrence based on known factors that affect vegetation. And then once you subtract that, you add in a deposition to see how much deposition may be playing a role. And they've been very useful, as you see, across the United States. Chris and Todd have all been doing these. and done a fantastic job. I'm part of the CLAD and a little bit envious of what you guys do in the States compared to in Canada. We don't quite have it as coordinated, I'd say. But there are limitations to this approach in that oftentimes you don't necessarily understand the mechanisms. So you have observations, statistical modeling. While we believe we know what's going on, sometimes we really don't. We've heard a lot about the reductions in emissions and deposition across the United States and how good it's been. Across Canada, it's pretty much the same. Nationally, emissions of sulfur and nitrogen, or oxides of nitrogen anyway, have gone down. But there are areas that are sort of bucking the trend. We're seeing increases, and I happen to work in a couple of these areas. One of them is the oil sands in northern Alberta, which you can see on the map there. And it's a little bit different the way critical loads are going to be applied here, or they want to apply critical loads, in the sense of it's a point source. You're trying to manage a localized emission source using a critical load concept, which is quite interesting and challenging in some ways. Initially, it started off as a NOx SOx, and the primary concern in kind of 2004, 2005 was the acidification. And you can see in the black there, the sulfur deposition was high, but it has dropped off quite remarkably over the last decade or so. In contrast, oxide emissions of nitrogen have continued to increase. So we're shifting more to the concerns over nitrogen as opposed to sulfur. A little note there that this is a dry part of the world. The annual precipitation in Fort McMurray averages around about 380 over the last 30 years. It can be as low as 240 millimeters. So that may become important later. So very briefly, don't go walking in the woods in northern Alberta because you'll meet some crazy people. But we conducted a 5-year experiment where we applied nitrogen as ammonium nitrate as a fine mist above the canopy. There's not many of these kind of studies that have been done. And this was done at the request of industry. We could have just done a critical load the way we've been doing it and said, this is it. Boreal forest, 10 kilograms nitrogen per hectare. Don't go above that. But they weren't happy with that. They wanted to know what's going on. How does it affect the environment? We want to know. So this was directed at a process-based. It's unusual in the sense that it was above the canopy. We applied deposition rates that were within the observed range in the oil sands. So our maximum deposition was only 25 kilograms. We went in small increments, 5 kilograms. That's because a criticism of many studies is that you add too much nitrogen, and it's not realistic. So we tried to do that. So we set up these plots at 6 treatments, and we applied it 5 times a year during the growing season. We got up at 4 in the morning. The students got up at 4 in the morning, went to the forest. When it was very little wind, we had spray paint, so we made sure that the deposition it worked quite well. And then we measured everything. We go out and so we had 2 kinds. We had destructive plots where we were interested in processes. How is nitrogen behaving in this system as we apply it? And so you can see we measured things like, obviously through fall, stem flow, we measured lidofall, soil veg chemistry, nitrogen processes in the soil. Even some years we did enzyme activity, ammonia homogenase to look at nitrification, fungal bacterial ratio. We tried to measure everything so that we could go to industry and say, look, this is what nitrogen's doing. And then the focus of this talk is we also measured just in monitoring plots, which were non-destructive outside. We didn't walk in them. We measured cover of, and I'm going to focus on vascular plants here in this talk. An interesting thing to note about this part of the world is that it's dominated by this reindeer lichen mat. It is crunchy when you're walking through, so you can hear the blackbirds coming when they're chasing you. You crunch, 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 crunch. So that's good. I've got another story about that if you want. Anyway, but we have the quadrats, and we measure the vegetation annually at something like 24 sites per subplot per treatment. I'm not going to talk about this, but we were applying it above the canopy. We did get some canopy interaction, more so for ammonium as opposed to nitrate. So we did get some of the applied. So deposition rate is above the canopy. Not all of that nitrogen gets to the forest floor. So that's an important thing when we're doing critical loads, because we're measuring above canopy, not necessarily what's reaching the forest floor. Some epiphytic lichens, which these systems have a lot of, nitrogen concentration increased with deposition rate. So, and we also got a small increase in organic nitrogen at the higher treatments, again, showing canopy transformations are occurring. But by and large, about 70-80% of the nitrogen that we applied reached the forest floor. So when we went into this, what we're expecting to see is a change in vascular community response with treatment, and that we would see the high nitrogen depositions would start to change the composition. But we didn't see that. If you look at the left figure there, total species, and this is all normalized to the initial cover or richness when we started the study. And you see this parabolic trend where, you know, in 2012, 2013, you got higher species, mostly of the common species. But it was in the low treatments, in the control and in the 5 kilograms. At the higher one, we didn't really see that. And by the time we got to 2015, 5 years after the experiment, we're back to where we started. So nothing happened, it's all good. But something did happen in the meantime. And so that's really what we were trying to understand, well, what was going on there? So we tried everything to look at. We looked at nitrification rates to see if this could, and the only thing that we could really see that explained the change in the vascular response was summer precipitation. That both diversity, richness, and cover was related to summer precipitation in those years, irrespective of treatment. It just so happened that the lower treatments, the response was higher. So we have this observation. In the end, so what do we think is happening? Well, as I said, we measured pretty much everything you could measure to see that, and no mineralization changes. We had lysimeters in there. We had no nitrate leaching. The vascular plants did not show an increase in foliar nitrogen. The lichens, this lichen mat, that's where it went. It really accumulated, and we even divided lichens into the apical, the necrotic, and then the fibric as it starts to enter the mineral soil. We did a N15 addition, which kind of showed that, it enters the system. Actually, interestingly, the nitrogen 15 did get into the vascular plants. So we didn't see an increase in concentration, but when we add a tracer, we know. So basically, the nitrogen, what we're adding, becomes part of the nitrogen cycle. But it doesn't increase the availability to vascular plants. It all goes into the lichens. And this is, if you look at it there, just looking at the deposition, and you convert, rather than concentration, you go to loads. Most of the nitrogen that we deposit goes into this thick lichen mat that you can see there. And what we tried to do then, and I did it for all treatments at the bottom there, we tried to forecast, well, what's going on over time, because again, a drawback of a study, it's 5 years, and it stops just as things get interesting. But that's where we're at. So we have this thought about, ultimately, of course, it can't continue to accumulate nitrogen. It's going to saturate out, and it will go somewhere else. And what happens at that point right now is somewhat of an unknown. So this is our best guess of what we think is happening to the nitrogen behavior in the system, is that the nitrogen gets deposited. It's taken up primarily by the lichens. It gets internally transferred a little bit because nitrogen, they don't have roots, but they can transfer nitrogen within the system. Over time, it's organic nitrogen, right? So there's no nitrate, there's no ammonium. It's getting converted into organic forms of nitrogen, and that's getting transferred into the mineral soil. These mineral soils are very low in nitrogen because they're a forest fire system. Every 60 or 80 years, we get a forest fire. We lose nitrogen. So the nitrogen concentrations naturally in these soils are very low. They're very sandy. So we're starting to see organic nitrogen, organic matter accumulate in the soil, but we're not seeing any enzyme activity, no detectable ammonium nitrogenase. So there's no nitrification. We got next to no net nitrification rates, no nitrate leaching. It's basically going into the mineral soil, modified by this thick, this thick mat. So why, getting back to the response to the vegetation, and what do we? I could be completely wrong, by the way. This is just a hazard of guess. So what I think is happening is that when we have low nitrogen treatments, The lichen mat and the vascular plants, there's no real change. So when you get more precipitation, they're equally competing. And because it's a water-limited system, there's more water for the vascular plants. Therefore, relatively speaking, they do better in those years at the low nitrogen treatments. When we add nitrogen, because these lichens are grabbing it all, that will likely promote photosynthesis and water use. So the idea is that the lichens may have a competitive advantage because they've got the nitrogen, they can photosynthesize at a greater rate, they're going to use the water, and therefore, in that case, rather than kind of being equal balance or more or less between the vasculars and non-vasculars, the non-vasculars are actually using more of the water. So there's less of the water that's available to the vascular plants, and that's why we don't see as much as a positive response to the higher precipitation. That's the best guess I have right now. And to conclude, just so we did this 5-year study at the request of industry to look what's happening, and then it stopped. Have we got anywhere? Well, we also have some sites where I've done some monitoring, and we have one in the left here that is very close to the Syncrude open pit mine. Mordor is another word for it, if you ever go there. And the one on the right is our study site, which is quite a ways from the site. And you can see just looking at it, it's quite different, right? There's a lot more green, a lot more vascular vegetation at the site very close versus the one further away. So when you actually do just a comparison, now this site has had about 20 kilograms of nitrogen for about 50 years, we estimate. And in this case, you see a very different response than we got from our 5-year experiment. The vascular biomass is about 3-4 times higher at the high deposition sites. It's dominated much more so by vascular plants, particularly Artostophilus, compared to the reference site, which is this lichen-dominated system. And given what I've just said about the importance of this lichen mat kind of regulating nitrogen behavior in the system, if you now start to lose this lichen mat, we expect that nitrogen is going to behave quite differently now once you've done that. Why we've lost it? I mean, I'm not saying it's nitrogen because in this site we have multiple pollutants. It's not just nitrogen. We have a lot of dust, a lot of other chemicals going on. So what's actually responsible for the different observations, I can't tell you. And that's, again, it's this trade-off between observations and experiments that I started with. You can see it in effect, but trying to explain it in these experiments, but sometimes experiments are not long enough for you really to get at the other side of things. So, take-home message. After 5 years, pretty much nothing happened, or very little happened in terms of nitrogen. So, at that level, you could say 25 kilograms is fine. And that's a problem, given the fact that what we've just seen with the observations. So it is a limit of an experiment. We have learned something about the way nitrogen behaves in these environments, which is very different, certainly to most places in the United States, where you don't have this lichen cover, that you do get a lot of regulation. So the inorganic nitrogen gets taken up very rapidly, converted to an organic form, and therefore not necessarily leaching or doing some of the other effects or availability to vasculars. There does seem to be a link with precipitation. It's an observation we saw, a mechanism we don't know. I'm hazarding a guess that it is a competition effect and that the non-vasculars are out competing. Can we use this to forecast, Evan? No. I mean, what we've seen is not right now very useful for us to forecast the critical load data because the experiment stopped after 5 years. And there's nothing from our experiment that we can use to tie into those observations at an actual quota, maybe because it's multiple pollutants, as opposed to just a singular pollutant. And that is it. Thank you.
Do we have questions for the speaker?
Thanks for this great talk and those great measurements. So I'm curious about, I don't know if you did any measurement within the canopy, because there is a lack of understanding of nitrogen exchanges within the subcanopy airspace. So, and the other question, so how deep actually, what's the soil depth? Because I think you sampled, you had soil sampling, right? So what is the depth of your soil measurements?
Okay, so for the canopy, we measured foliage. With jack pine, they have multiple years, so we split them up into first year, second year, third year needles. We also measured lidofol. We also measured epiphytic lichens, the two most dominant lichens. We didn't see anything in the jack pine foliage, both in lidofol, retranslocation, first, second, third year. The trees didn't seem to get it. The one thing that we missed was the bark, the actual branches, which were dominant there. So some of it may well have gone there, but it certainly went in the epiphytic lichens. Concentrations went up by 30, 40% in the areas we sampled. These soils, I've dug many soil pits in my life, and these are the best. It's like Blackpool Beach, if you've ever been to Blackpool. It's just sand, right? Not a rock to be seen. And so actually, it is an interesting... we have lysims were at 30 centimeters depth, but when we measured the soil, we separated it by horizon. So we had the AH horizon in Canadian soil classification, and then the upper B horizon, as well as the forest floor, the FH and the lidder. Actually, it's quite interesting. If you go a meter, so we'd put lysimeters in not this place, an adjacent site, and in spring, we only got water in our deep lysimeter. We didn't get water in our shallow lysimeter, which, we know water goes downhill. So how is that possible? And the reason is, actually, it's a very flat landscape. We're very, very close to the water table. So I think what's happening in the spring, fresh air, the water table is rising, and our deep is just above the water table, and it's actually collecting this groundwater. So we were well above that where we were measuring.
So you're considering only the aerobic processes for the nitrification, not the anaerobic ones. You didn't go very deep, right? Concerning the water versus the nitrogen and the nitrogen cycle as well. Did you consider that in your analysis or not?
Sorry, we didn't consider what, sorry?
Sorry, for the nitrogen process is the nitrification.
Yeah.
So it's only in aerobic conditions, right? So you only consider that in your analysis?
Yeah, because these are sandy soils, they do not get saturated. There's no anaerobic. You never get standing pools. As I said, it's pure sand, almost. So that's why they're very sensitive. We also did net nitrification, so we have buried bags to look at that, and we didn't. I mean, that was done monthly, and we pretty much very rarely got nitrate measurable.
Okay, so I'm pleased to introduce the next speaker, who is Jennifer Olguin from the University of Texas at El Paso.
Yes, good morning, everyone. My name is Jennifer, like Jenny said. I'm a PhD candidate at UT El Paso, and today I'm going to discuss findings from a 4-year realistic nitrogen deposition experiment that we conducted at Carlsbad Caverns National Park, and how our responses may have interacted with extreme climate events. So as many of us know, nitrogen deposition rates have declined or plateaued in some parts of the world. And in the US, policies such as the Clean Air Act amendments of 1990 helped to drastically reduce nitrogen deposition through the reduction of nitrogen oxides. However, despite these dramatic declines, nitrogen deposition is still estimated to be 5 times above the pre-industrial levels across most of the US. And the critical loads, or the level of deposition below which no harmful ecological effect occurs, has long been exceeded in the US, particularly so in the eastern half. And these exceedances are likely to remain so under current policies. The vulnerability of ecosystems to nitrogen deposition is not unique to the eastern half of the US, however. In 2016, Sullivan and colleagues released a report evaluating the nitrogen critical loads and potential exceedances in 15 national parks. And these parks were chosen because they were identified as having nitrogen-sensitive ecosystems, and some, including Carlsbad Caverns National Park, were experiencing increased levels or exposure to nearby nitrogen emission sources. And I'm going to start calling Carlsbad Caverns National Park Cave because it's long. And in this report, they showed that nitrogen critical load exceedances was within 1 kilogram of nitrogen per hectare per year over vast portions of cave. However, the authors did note that the data reflected estimates from 2011 and thus may not fully capture future emissions and deposition if they had changed. And emissions changed. Between 2017 and 2019, U.S. oil and gas production volumes had grown up to 100% in key production areas. And in the Permian Basin, where cave is located, oil and gas production volumes had doubled, and the nitrogen oxides emissions had shown to be the largest among all of the production areas assessed. And hopefully you can see that clearly here. Determining the critical loads of nitrogen in ecosystems across the U.S. has been a forefront of research over the past decade. However, there's a clear gap in knowledge in the southwestern part of the U.S., where most of the desert ecoregion data has been acquired from the western section of the Sonoran Desert ecosystem downwind of Los Angeles, which has seen a lot of deposition. And this gap is particularly important because of the challenges associated in predicting the impacts of nitrogen deposition in arid lands in general. For instance, in the literature, you'll find that some arid land nitrogen addition studies show strong responses to nitrogen additions, while others report little to no response. Conflicting responses between studies may be due to a variety of reasons, such as differences in experimental manipulation, so like the amount of nitrogen that they're adding, how long the experiments are running, even the frequency of nitrogen throughout the year that they're adding it in. And maybe perhaps even more importantly, there is high spatial heterogeneity across these arid land landscapes due to factors such as differences in geological history, physiochemical factors, and even plant communities, which can show differing sensitivities to nitrogen. And of course, since we're working in arid lands, we also have this looming and often unpredictable water limitation that can essentially dictate whether or not we see a response to nitrogen enrichment. For instance, in this study by Ladwig and colleagues, they only found a response to pretty high levels of nitrogen deposition or addition over 10 years following a prescribed fire and well above average monsoon season. And the role of water limitation can be particularly problematic for regions that are expected to experience changes in precipitation patterns or amounts, such as changes to the North American monsoon, where during this period, Mexico receives most of the rain, but there's parts of Arizona and large parts of New Mexico that receive more than 50% of their mean annual precipitation during this period. The North American monsoon is predicted to undergo some changes. However, there is some strong disagreements in the models as far as the magnitude and direction of the change, where some models predict little to no change in total summer precip, while others predict increasingly extreme and irregular rainfall events that deliver less rainfall overall, and even pre-monsoon hyper-arid periods extended. So while we did not set out to do a nitrogen deposition climate change combination experiment, Mother Nature said otherwise. Nevertheless, the overarching goals of this research was to determine how realistic nitrogen inputs impact plant community structure and growth, as well as some biogeochemical factors in the Chihuahuan Desert grasslands of Carlsbad Caverns National Park. So this is just a brief site description of cave. Like I said, there is a very large oil and gas presence. To the left, you'll see all of the active new wells, everything. The park is located in the southeastern region of New Mexico in the Chihuahuan Desert. It's classified as semi-arid. Most of its rainfall falls during the North American monsoon season, and the critical loads for vegetation was estimated to be from 3 to 8.4 kilograms of nitrogen per hectare per year. Our experimental design was designed around the model deposition that was predicted to be occurring at the time of the study design. It was modeled to be occurring in a gradient from 3.5 to 4.5 kilograms of nitrogen per hectare per year, and we established 3 sites within the lower part of the gradient. And we had 4 levels of nitrogen addition treatment. The first was ambient, so we applied nothing. It's assuming background deposition. And then we had a nitrogen reduction treatment where we added a labile carbon source that would induce nitrogen immobilization by microbes. So basically they take up the nitrogen into their bodies and it's no longer available to the plants. And that's to assess if this ecosystem was already nitrogen saturated. And then we have a nitrogen low and nitrogen high addition treatment which equated to 6 kilograms and 8 kilograms of nitrogen per hectare per year. And this was applied once annually as ammonium nitrate. These are the 3 grassland sites. 2 of them were dominated by perennial native grasses, and 1 of them was dominated by a perennial exotic grass, Erigostis lamania. I do have a chapter dedicated to figuring out if there's an interaction between the presence of this invasive grass and nitrogen enrichment, but I'm not going to go over that in this presentation. So yeah, you can ask me later. The soil texture among all the sites, soil texture is extremely important as far as biogeochemical cycling goes in the desert. So they were all similar across sites, and we had similar soil pH and nutrient concentrations before the start of the experiment. For the first 2 years, summer rainfall was well below the 30-year normal, where 2018 was only 50% of the normal, and 2019 was only 46%. So we decided that just in case nitrogen addition effects depended on water availability, we decided to add a water addition treatment for the next 2 years at one of the native sites to compare the 2 natives. And our aim was to increase monsoon precip by 30%, assuming we had a normal monsoon. And yeah, we didn't. We had a really worse monsoon season, and it was hotter. This is just a visual of how 6 water additions in, we were watering crispy grass the whole season. So we fully intended on going out in 2021 to add water to the site again, but we had some crazy early monsoon flooding that basically prevented us from accessing the site at all. Yeah, but it was okay because that season we got about 25% of the normal, so it's a high monsoon season naturally. So now I'm going to go over the nitrogen input effects on plant community growth and structure. Total relative percent vegetation cover, we only found treatment effects during the first year that we sampled, and the treatment effect was the percent cover was highest in the ambient plots versus the nitrogen low. For the cover of plant functional groups, again, we also only observed treatment effects during 1 sampling period, so 1 year. And this was during the final year where we had most of the rainfall, or the highest rainfall amount. And I'm not going to get into the nitty-gritty of the treatment effects, but I just want you to see the pattern. You'll see. So the perennial grasses, we only found effects at 1 site during 2021, perennial forbs at the 2 native sites, and annuals, we did not find any treatment effects. But as you can see here in the orange, we did get quite a bit of annuals after the extreme drought in 2020. For diversity measures, we found, again, no treatment effects, but unsurprisingly, we did find some sampling effects, where diversity was lowest during the really bad drought and highest during the high rainfall year, except for one of the sites that had a pretty good recovery of perennial grasses, grasses. For plant species composition, again, no treatment effect, but as you can see clearly, we have quite a bit of difference of the 2021 high rainfall year from the previous years. I couldn't plot 2020 because it's just nothing but zeros. So our highlights for plant responses: treatment effects were scarce and inconsistent, and we only observed responses during the first or the final year. And the 2020 drought likely drastically reduced, it reduced total plant cover and likely led to the significant shift in plant community structure where we saw a ton of annuals pop up. Now I'm going to go over the effects on biogeochemistry. We measured quite a few things, so from soil pH, nutrients, extractable organic carbon, total nitrogen, microbial biomass, and carbon, nitrogen, and phosphorus activities, and some other stuff that I didn't want to stuff into this. But as you'll see here, a lot of the treatment effects occurred during a specific season and year. But like the plants, the treatment effects were often weak or inconsistent, and I'm not going to go over all of the treatment effects, but I'm just going to give you a little taste of it. For available soil nitrate, you'll see that treatment effects only occurred during the winter of 2019 and 2021, so the first and final winter that we measured it. But you'll also see that the treatment effects were short-lived. So after the winter season, I sampled the pre-monsoon and monsoon season, so we didn't observe any treatment effects after they showed up. For microbial biomass carbon, this indicates microbial limitation. So if they're limited by nitrogen, they'll grow more. We did have a treatment by sampling effect, but post hoc differences did not show anything, and I'm not seeing that strong of a difference visually. And this is in contrast to an incubation experiment that I conducted before the start of this experiment, where we showed that these microbes were limited in the perfect conditions. They had all the water, no plants to compete against, and whatnot. So just to recap, nitrogen effects on soils. So soil nitrogen and microbial activities were weak and inconsistent and often short-lived. Responses also strongly depended on the season and year that we were measuring. So drought or not, we've been applying nitrogen for 4 years. Where is it going? It could have accumulated in the soils, but I wasn't seeing I wasn't convinced. Or even if it did happen, it wasn't long-lasting. Could also be going to plant biomass, but remember we did find some weak and inconsistent responses. I do have plant tissues that we collected in the final year, but I'm still working on the data for that. We could also have some leaching where nitrate is pretty mobile, but this is I think it's highly unlikely during the drought years where I didn't see any nitrate accumulation. We can also have some gaseous loss, abiotic gaseous loss, or even through microbial mediated activities like nitrification, denitrification. To test this, we conducted an experiment earlier this year where we collected soils after 4 years of treatment from the ambient, nitrogen low, and nitrogen high plots. And in the lab, we added either deionized water or more nitrogen, and that equated to the highest fertilization treatment in the field. And we incubated for 1 or 24 hours at 20% water holding capacity. And we measured nitric oxide and nitrogen dioxide. So preliminary results for this study: after 24 hours, soils that were treated with nitrogen in the lab released more nitric oxide than the water-only treatments, regardless of field treatment history. And just as a note, beginning soil nitrate and ammonium nutrient concentrations did not differ between the soils that we collected. Some overall key findings: the nitrogen field treatment effects were weak and inconsistent. And because treatment effects often occurred during the first and last year, I would say it's unclear whether the system is sensitive to nitrogen or not. And also, soils did show strong potential for nitrogen gas loss, primarily as nitric oxide. So now for some future considerations for nitrogen deposition research in this region. U.S. consumption and production of petroleum and other liquids are projected to grow through 2050. And while there is strong disagreement in what climate change will do in this region, empirical evidence does show that aridity in the Southwest is increasing, and there's more. We have evidence of more prolonged and severe droughts that are already occurring. And now some final thoughts. Ecosystems across the globe will likely experience multiple global change drivers, and the high heterogeneity between air-land landscapes may complicate predictions to global change drivers like climate change and nitrogen deposition. So we must consider long-term coordinated research across a diverse number of air-land landscapes to perhaps fully understand the vulnerability of these ecosystems to nitrogen deposition. These are my acknowledgments of all the amazing people that have helped me throughout this process, and the bats too. And just to leave you just to show that the crazy weather continues. This is in 2022, this year. Yeah. Thank you.
Do we have any questions for Jennifer?
Yes.
This is really great. I mean, one of my favorite things to see is that some of these new sources of nitrogen have no effect, and therefore parks are unlikely to be harmed in that space. But how do you think like a field campaign, like with these lab studies of looking at gaseous loss of nitrogen, like how would it be valuable to look into that, and what would that look like?
Huh. Wow. Yeah, I think it would be totally worth looking into, because I mean, it's releasing a very powerful greenhouse gas, the nitrous oxide. We didn't measure it, but I'm sure they might be. Nitrogen dioxide is also important for ozone, so I think it would be worth looking into how these gaseous losses could be contributing to even worse air quality and climate change, maybe. Thank you.
And I'll just make a plug for ammonia. If you do such a field experiment, include ammonia fluxes as well. In these higher pH soils, we still have a lack of understanding of what's going on with ammonia, particularly in these landscapes as far as air surface exchange goes.
Oh, interesting. Thank you.
Okay, if there's no more questions, then we'll move on to the next talk. Okay, and for our last talk of the session, we have Patrick Levasseur from Trent University.
Hey guys, how's it going? So anyway, like we just said, my name's Pat Levasseur, and I'm a PhD student at Trent University in Peterborough, Ontario. And today I'm going to be presenting actually the honors thesis from a student in our lab, Jody Newman. Jody's not here today, but she's on the internet, I think, right now. Hi, Jody. The title of this presentation is The Impact of Severe Pollution from Smelter Emissions on Carbon and Metal Accumulation in Peatlands in Ontario, Canada. Okay, so peatlands are a wetland ecosystem where waterlogged conditions prevents the full decomposition of plant material, resulting in the accumulation of peat. And the rate of peat accumulation can be influenced by environmental factors, such as temperature, water table height, and type of vegetation. And although peatlands cover only about 3% of the global land surface, they contain around 25% of terrestrial stored carbon. So these are massive carbon pools that, as we focus more on climate mitigation and and increasing carbon sinks, peatlands are a major factor in that. And so not only are they massive stores of carbon, but they also play a large role in water filtration, erosion control, and flood prevention. They're also biodiversity hotspots, as most wetlands are. And so there's a picture of Jody. So this manuscript's actually under the review process in environmental pollution right now. So in northern peatlands, they're often dominated by sphagnum, which is a genus of moss that's considered an ecosystem engineer due to its ability to manipulate hydrological conditions to favor peat formation. Sphagnum decomposes very, very slowly, and so it is generally how peatlands are formed is using sphagnum species. So I have a picture there. With the mouse? Yeah. So anyway, down here is a species of sphagnum fuscum. That was quite common at some of our peatlands. And up here is a picture of a bunch of different types of sphagnum species that we found all at one of our peatlands. But sphagnum is also quite sensitive to atmospheric sulfur compounds, and we've seen in lots of areas that have high sulfur deposition that sphagnum, like a lot of other non-vascular plants, will die quite quickly with elevated sulfur concentrations. And the loss of sphagnum from peatlands can increase peat decomposition. Pretty much the fresh sphagnum layer on the top acts as a cap preventing the rest of the sphagnum from reaching exposing it to oxygen. And as soon as the sphagnum layer is removed from the top, it increases decomposition as the decomposing sphagnum underneath is exposed to oxygen. And so this decreases peatlands' capacity for carbon accumulation. So peatlands are also, they've been used as pollution archives for quite some time. They've been used as temporal records of metal pollution across the world. So this was a paper that was published in Science in 1998, looking at the history of lead pollution for the past 12,000 years. So you can see on the left here is the enrichment factor. That's weird. I can't see the mouse. So, and so you can see when humans first started mining lead, the Phoenicians and the Greeks here in 3000, you can see we have a peak in the enrichment factor for lead. And then, anyway, the scale changes dramatically. And you can also see quite clearly here when the Industrial Revolution began using lead enrichment factors in peatlands. So, and the concentrations of these metals can be measured and combined with radiometric dating techniques to construct a pattern in metal accumulation. However, these studies often assume that metals are immobile in peatlands, that they don't fluctuate up and down a peat core. And the affinity of peat for different metals can be affected by pH, the vegetation type, and impacted by processes such as drought and heavy rainfall events. And we're starting to see more and more that different types of metals have different affinities for peat and potentially change along a peat core. So our study is going to take place in the city of Greater Sudbury, which is in northern Ontario. Ontario is a really big province. It's not, anyway, if you look at the map, it's not even in the middle, but it's considered one of the capitals of kind of northern Ontario. So Sudbury is famous for large-scale mining and smelting operations that began in 1888. And in the 1960s, Sudbury was the largest point source of copper, nickel, and sulfur dioxide emissions in the entire world. And so to fuel a lot of these open bed roast yards, they harvested the whole area. They cut down all the trees, and then acid rain and metal deposition was so high that no vegetation could grow back. So it led to 20,000 hectares of barren land, including highly degraded peatlands where lots, if not all, of the sphagnum species were completely removed. So there's a picture of one of the old roast yards. And I included another picture just to kind of, not only did it kill all the vegetation in the area, but the microbial communities were also really devastated. So you have these kind of ancient stumps like these ones all around the city still that just haven't decomposed at this point. So it's a pretty dramatic landscape. However, it's also home to one of the world's largest regreening programs. Sulfur and metal emissions have decreased by, I think, over 98%. And the current air quality in the city of Sudbury is within all the Ontario guidelines and is equivalent to a day in Toronto. So you can see here the drastic reduction in sulfur dioxide emissions. And so along with the reduction in emissions, they've also had applied crushed limestone and fertilizer to 3,400 hectares of land. They've also planted around 10 million trees. This really is one of the world's largest regreening projects, following quite a bit of environmental devastation. So it makes for a very interesting project, a very interesting place to study pollution and peatlands response to pollution, as the area is quite dominated, well, it was quite dominated by peatlands, like a lot of Northern Ontario. So the widespread degradation of peatlands in the Sudbury region provide a unique opportunity to evaluate whether severe pollution and associated loss of sphagnum impacts carbon accumulation rates in peatlands. And given that the two major metals that were emitted from the smelter, copper and nickel, exhibit different geochemical behaviors, we were also going to look at how copper and nickel move up and down the peat profile, or if they do. And so to do this, we were going to do radiometric analysis of peat cores from 3 different peatlands, ranging in distance from a smelter. And we were going to use those peat cores to test the hypotheses that carbon accumulation rates will increase with distance from the smelter, and that peak peat metal concentrations will coincide with the time frame of greatest smelting activities. Oh, well, I just wanted to highlight that's a purple pitcher plant right there, triple P. Anyway, it's a very nice, very nice plant that you see in a lot of the peatlands in that area. So these are our 3 different peatlands, and like I said, they ranged in distance from the smelter there. So on the left, they were all named after the lakes that they were attached to. So on the left there, we have Silver Lake. If you look in the background, you can actually see the superstack there, and this is the highway right here. So this is our site that's quite close to the smelter, and then we move further away. Clearwater Lake was 12 kilometers from the smelter, and then we have Cartier Lake, which is 41 kilometers from the smelter, and this was thought to be relatively unimpacted by emissions from the smelter there. And that's, anyway, as you can see from the picture there, that's what you'd expect from it looks more like a northern peatland than the other 2. So this is just a map of the study area. The red X's are our 3 study sites. So again, yeah, that's Silver Lake, closest to the smelter, Clearwater Lake, and then Cartier Lake is way over here. So, but along with our 3 peatlands, we also had data from a previous study done by Suter and Watmow, and they looked at 25 different peatlands in the area, and so we were able to use some of their data for this study as well. So peat coring and sample prep. So we took one 50 centimeter peat core from each of the peatlands. There's a picture of me and Jody pulling a peat core out of the ground. So we take a PVC pipe tube, and you hammer it into the ground, and then you got to stick your arm down and cup it on the bottom and pull the whole thing up. It was very helpful to have. Jody has tiny little arms, so it's very easy to punch through the peatland and lift it up. That peat core we actually named Peter Corey. We're very original and funny. But you have to make sure when you're hammering the peat core down that the top of the core lines up to the peatland, to the peat outside of the core, just to make sure that you're not compacting it. And then we took each one of those cores and we sliced them into 1 centimeter increments back in the lab, making sure to clean the knife in between one of those cuts. And then each of those samples were dried in an oven at 100 degrees Celsius for 24 hours. And then we took bulk density measurements by weighing those samples, and then they were all milled using a coffee grinder. So there's a picture of our peat core, some of our peat samples out for analysis. So we took we conducted a lot of different measurements: pH, carbon, nitrogen, and then the concentrations of copper, nickel, lead, zinc, and titanium. And then we also sent these samples out for lead-210 dating at the University of Ottawa. And so lead-210 can date these cores, date peat formation back 150 years, which coincides with when smelting began in Sudbury. So, okay, yeah. So these are the picture of our 3 different cores. So you even just visually, you can see the difference between the 3 of them. So particularly the Cartier sample on the right there, you can see it has that light fluffy sphagnum on top. and kind of gets darker and denser as you go down. Whereas at Silver Lake and Clearwater, pretty much right at the top of those cores, it's incredibly dense right away. There's effectively no sphagnum cover at either of those two sites, 1 to 3% cover sort of thing, whereas at the Cartier site, it's 80% of the site is dominated by sphagnum. Not only one sphagnum species, but that picture there was all of the 8 different sphagnum species that we found in a very small site. Anyway, that's, again, just a different picture of them. So you really have, anyway, so even just looking at these, you can see there's quite a drastic difference between these 3 different cores. And so using the lead-210 dating, I'm glad to see that lead-210 got back on its feet again in its dating. Again, I said I was funny. There we go. I'm the first one to ever make that joke. So you can see the difference. The values on this figure here are the earliest dates that we could get using the lead-210 dating. So you can see again at Silver and Clearwater Lake, those 1858, 1832, that's only at 10 centimeters deep in those peat cores. Whereas at the Cartier site, it's much, much deeper. Like 35 centimeters deep, we got down to 1842. So the peat accumulation just using depth is much, much larger at the Cartier site compared to the 2 that are close to the smelter. You have 180 years of peat accumulation just in that top 10 centimeters. So, and then looking at the metal profiles, so copper and nickel are the 2 primary metals that are emitted from those smelters, and we wanted to look at how those lined up with the lead-210 dating and the ages. So again, these are 3 different peatlands. So you can see here that the highest points of, so the red line is when, I can't see it on that.
There's a pointer on that other thing. If you click up that and then the top red button.
Okay, it's pretty little, but okay. But anyway, so this red line here is when smelting began. So you can see obviously that changes again just at the depth of those peatlands, and then the green is when the sulfur emissions total emissions decreased in all of these areas. So you can see that. So the solid line up here is copper, and the dash line is nickel. So you can see the copper. It's super distinct, right? These copper concentrations really happen right around when the smelter emissions occur, and there's almost none before. Whereas the nickel, there's quite a bit of smearing going on. We have, although it's most elevated at this point, it's not quite as clear, and some of the nickel that was deposited during this time frame is moving up and down the profile, which is not as true for copper. Zinc and lead, we also looked at these. These weren't smelter emissions. These are more from leaded gasoline and a bunch of other reasons, but these weren't point sources. And so we found similarly, and lead is often, like I showed earlier, lead is known to be highly immobile in peat cores, and we found the same thing, that that lead was elevated at very distinct points, whereas the zinc, there's quite a bit more smearing going on with the zinc. And so that's just with total concentrations, but when we used enrichment factors, we found the exact same thing. So what does that mean for overall accumulation rates? So despite the fact that the depth of peat was much deeper at the Cartier sites, because that upper layer for Silver Lake and Clearwater Lake was so dense, what it means for total carbon accumulation is that the values were actually quite similar. There wasn't a big difference in total carbon accumulation per year. There we go. Between the 3 different sites, despite the fact that we're talking 35 centimeters versus 10 centimeters, just because the density differed, it didn't actually make a difference for carbon accumulation, which was really interesting. However, for metal accumulation is what we expected. Those sites that were really close to the smelter had much higher rates of metal accumulation compared to the other ones. And then comparing that to the sites from Suter and Watmo, they found, anyway, we we found quite similar trends that matched up. Just using distance from smelter, again, the bulk density decreased as you move further from the smelter. You get more sphagnum species. It's much lighter and fluffier than what was happening close to the smelter. And anyway, so you can see that matches up with sphagnum frequency and copper and nickel concentrations in these peak cores as we move further away from the smelters. So I'll just wrap up here. So despite extremely high pollution inputs and loss of sphagnum, carbon accumulation in the Sudbury peatlands has been relatively unaffected from this centuries of intensive pollution. And the copper and nickel were greatly enriched in surface peat at the two closest sites to the smelter. And metal accumulation rates reflect the declines in smelter emissions since 1970s. And copper and lead matched profiles of the smelting activities much better than nickel and zinc. So this project was part of a larger project, the landscape carbon accumulation through reductions in emission. So just wanted to thank all of our sponsors, and the City of Greater Sudbury, Province of Ontario, NSERC, Glencore Valley, and the rest of the LCARE team. Thanks.
Do we have any questions for Pat?
Thank you. A really interesting talk. I wonder if there's a strong prevailing wind direction that is associated with this distribution of the heavy metals.
Yeah, absolutely. So the Sudbury area, the emission rates were so high in that area. Everything within that region, the Barren Zone, was heavily impacted. Wind direction could absolutely have played some role, but that area that we were studying, that they refer to the Barren area, before the regreening project began, there was no vegetation left in that area, and the pH of all those soils was below 4.2. So we know that, aside from that Cartier site, which was outside there, all of those sites were heavily affected. The magnitude could have been different, for sure, but we know that those sites were heavily impacted. Good. Okay. Thank you very much.
Okay, so NADP had a committee. It went out there and it looked at all the various ways in which other people around the world were looking at this issue. Canada cropped up as number one. Could we just simply adopt the CAPMON procedures? So we spent a year and looked at those devices that they were using, filter packs. And what we found was that the pressure drop involved in them and the humidities that we find in Tennessee, in particular and south of us, are such that you could expect that when you cracked one of those open, you might find liquid coming out. In other words, the filters were getting wet, and hence the suspicion was that sulfur was being reported as sulfate and not as gaseous SO2. So we said, we will not use that. We'll use something, we'll put a heater on the front end. We'll limit it to just 5 degrees Celsius, but that's enough to get past the diurnal humidity cycle in the southern United States. So we put that in place, and we started up a program looking at it, and we had that going ahead. Look, I was at that time involved in the National Acid Precipitation Assessment Program. We had several things going on in parallel. We had NADP wanting a dry deposition program. We had NAPAP endorsing what NADP wanted to do. We had Congress saying, hey guys, that's fantastic, phenomenal. And when NAPAP put it in its budget, lo and behold, Congress added money to the NAPAP budget. Congress decreed that the money be disseminated by EPA. In the corridor, Jackson Square in Washington, the head of the EPA at that time, who was instructed to handle this money, and I had a meeting. The question to me was very simple. Can you give us dry deposition measurements alongside what NADP is doing for wet deposition? I said, the science is not there, but we have a program to step forward. The response was, in that case, I will give the money to somebody who says he can do it. All else followed. That's the reason you haven't seen me at too many of these meetings. I got thoroughly and absolutely pissed off. But we went ahead with what the NADP was suggesting anyway, on a wing and a prayer. And we made this program, which had sites at different levels of complexity in different places, and we stepped ahead with that for many years, and we made some breakthroughs, yes. We got measurements of sulfur where we didn't think we could in the past. We went on to the NOx and finally to NOy. And we developed the multi-layer model. That was developed as a part of the NADP program in its early days. By early days, these are not early days for me. I'm talking about 1980s. That's not early. I have learnt that the way to handle, the way to get success in the scientific environment is to outlive your critics. This is what we have, or my depiction of it. I don't want to pay attention to the details of this. I'm just throwing up the fact that we have a hell of a lot of sites where good measurements are being made. This is great, fantastic. But I'm now promoting that we could go out there to a select number of these and actually calibrate them. Look at this. I'm changing eddy correlation. I'm saying that if you have a sensor that is operating so poorly that you don't trust it for standard eddy correlation, instead of relying on it absolutely, accept the fact that the transfer of that trace gas or chemical species is going to be governed by the transfer of heat. So measuring the flux of heat is damn simple. Go out and do it. But as you're doing that, take that temperature signal you're using, plug that into where in that data recorder it says, I'm going to compute a covariance, and then also plug in that output from the lousy covariance sensor, the lousy chemical sensor. All right? So here, in red, we have the temperature data going in. In blue, we have what your sonic anemometer, I presume a sonic anemometer, doesn't have to be. And then independently you get that lousy C data. So come up with 3 different covariances, 3 different correlation coefficients. I'm expressing this in terms of the correlation coefficient because I believe in eddy correlation. You speak about covariance and what the hell, it doesn't matter. But the mathematics, and the story I want to leave you with, is that as I promote this, I don't know of anybody in the world who's ever done it. I'm promoting it, I'm throwing out at you guys. If you get the opportunity, test this out, because if it is right, then we have a method, because we can come up with the eddy flux that you're interested in. Very simply, as expressed there. Bear in mind there's an assumption. The assumption is that the correlation coefficient between your chemical and temperature is close to 1. Now, in all that's holy, that falls in the category of we hold these truths. It ought to approach that. Of course, it probably doesn't. And the question is, I don't have the answer is that I don't have data to test that out. People sitting in this room have data. So I'm throwing this up there in the hope that some of you will go away and say, look at what that lavish-ing idiot was telling us. And look at this. He was absolutely wrong. Well, could be. However, if we test it out and it proves that I'm not wrong, we have a method now to make lousy sensors useful. So what are these new sensors? Here's a variety of them. Some of them are solid state. There's a big unfortunate thing about the solid state sensors available today. Inevitably, they're made in China. Doesn't worry me, but I can see that maybe the availability might suffer in the near future. So, my claim, the technology is now here. New instruments are available. I think it's now possible to calibrate NADP sites, some of them, not all of them. By the way, I'm hyper aware of the fact that there's an ongoing dry deposition program here that is relying on using models to come up with these same data. I claim that their outputs are what the model tells us. I want to go out there and test the sites and see whether or not we can get absolute reference points. At that, I'll take you back to where I came in. Please think about the Schmidt number to the 2/3 power and think it through. I have not published this. Well, actually, it will be published early next year. I have a book coming out. It'll be in there. So anyway, thank you for this opportunity. You folk are doing fantastic work. Keep it up. And this, I think, is something that's worth testing. Thank you. And by the way, all of that initial work was done under NAPAP, and it's all described in the NAPAP Science and Technology documentation. If you don't have that documentation, I have 5 copies of it right here. Come and grab, they'll go fast.
Any questions? Or someone willing to stand up and take that challenge? Anyone?
Lunchtime.
Could be, could be. Anything online? Nothing online. Okay. Well, then I think we'll, you can catch up with Bruce after the session, maybe during lunch. You want to wrap up? No? I'm sorry? Do you want to wrap up or? Okay. All right, then we'll start up again on time at 1:30. Thank you again, Bruce.